Genetics that they'd like to ask before we start talking about membranes? I hope those questions will clarify and you'll take them to the TA's office hours and to mine, but for the moment we'll plow ahead with membranes. Okay. So it's a little bit of a switch of gears. I'm not quite sure why your textbook decided to put membranes after DNA and protein and have the enzyme mechanisms and kinetics between proteins and membranes, although admittedly enzyme mechanisms and kinetics are aspects of proteins, so maybe that's the rationale. In any case, coming to the membranes, we have sort of the third large group of structures or molecular assemblages that we need to think about. And membranes are really quite different in their organization from either nucleic acids or from simple pure proteins. They're proteins in membranes, but they take kind of a different approach, as we'll see. Membranes are important for a large number of reasons. So their functions include simply creating an inside and outside, being a barrier, being a selective barrier. If it's just nothing can come in, life would come to an end. Membranes are set up in a way that allows for electrical transmission, which is weird. Now, we're more or less used to the idea that nerves would be involved with electrical transmission, but it turns out that the kind of electrical properties that nerves have is sort of an amplification of the normal electrical properties of cells. Like so many things, nerves are showing an expanded version of something that all cells show to some extent. Membranes can accomplish transport. They can make it happen in such a way that things that shouldn't go through a membrane do go through a membrane. They can even create a gradient across a membrane. That's going to be interesting. Membranes are the location for a lot of signaling. A lot of the work by which cells find out what's going on or talk to each other is accomplished at that interface between outside and inside. And recognition. If you've ever thought about the fact that your bodies hold together, your cells can recognize where they are and who they should be holding on to. And that's not trivial. As in all of biochemistry, the structures of the membrane will reflect their functions. So I've talked about some of those functions of the membranes. We're going to see that the structural basis through which membranes can be self-assembling. There isn't some kind of little machine in there that turns out membranes for the cell. They're self-healing, which is obviously important to things like being a barrier. And they're semi-permeable. And this is just a little kind of a schematic of the sort of an archetypal little vesicle showing you that cells are capable of changing their interior environment in such a way that the interior of the cell is enriched for potassium and depleted in sodium, and that the fact that they maintain themselves that way allows selective flow of sodium and potassium to create electrical signals. Okay. So the common element to all membranes is a lipid bilayer. Try to have clear in your mind what is meant by a lipid bilayer. So in other words, it's made up of lipids, and there's two layers. There's a bottom layer and a top layer, if you like. I've seen a number of permutations that rearrange the parts of the phrase lipid bilayer or confuse the concept of a double membrane with a lipid bilayer. A lipid bilayer is one membrane. What the chloroplast has around it, what the mitochondrion has around it, is a double membrane, each membrane of which is itself a lipid bilayer. Now, what makes this lipid bilayer form is the fact that it's made up of amphipathic lipids. Now, amphi, you're familiar with from amphibians, animals that can live in water and land. Pathic, feelings, strong feelings like sympathy and antipathy. So amphipathic lipids have both feeling. They are both hydrophilic and hydrophobic, if you like. And because the amphipathic lipids that make up the membrane have this strongly different behavior at the two ends of the molecule, this is a spontaneous structure. No more information is needed than merely the composition of the lipids themselves to generate a lipid bilayer like this. Not all lipids will make bilayers, but the lipids that do make bilayers do so spontaneously without any further instructions. Because this is a non-covalent structure that has formed spontaneously, if something disrupts it, if you have something stick through it, if you put a pipette through it, if you put a thorn through it, that lipid bilayer will just flow around and anneal over it and close up with no trace left behind. It's self-healing. And what are these sort of properties that are going to make it possible for things to have that thing? Let's take a little look here at two fatty acids. So here is palmitate. You can see it's a long hydrocarbon tail with a carboxylic acid at one end. Here's stearate, a slightly longer carbon tail, again with a fatty acid there. The combination of those fatty acids with appropriate backbones is going to give you the specific kinds of amphipathic lipids that are going to be needed for membranes. But you can see how even in this condition, this is going to be an amphipathic molecule. This part is very hydrophobic, very nonpolar, therefore hydrophobic. This end is carboxylic acid and therefore about as polar as we're going to see in a biological system. You've seen that schematic lipid bilayer in a number of different diagrams, not just in our textbook, but probably for as long as you've been doing biology. And it's a perfectly decent representation for as far as it goes. That is to say, there are head groups that are polar, that are hydrophilic. There are tail groups that are nonpolar and hydrophobic. So that there's this hydrophobic interior here that's very much a sort of a lipid sloshing around thing. And then this outer layers on both sides that are very polar. This diagram is a more accurate representation of a lipid bilayer. In the last few years, we now have enough computing power to do things like represent all the atoms in a lipid bilayer if you take a small enough chunk. And that's what this lipid bilayer has been trying to represent, is just not just this generic model here where you kind of have squiggles, but actually atom by atom, what would it look like? And this is showing you the palmitic and oleic acids in here. Palmitic is, again, the straight chain hydrocarbon we talked about. Oleic is an unsaturated one that you can see some of the kinks in there. You can see that the nitrogens are focused out here on the outer faces, the polar domains of the membrane. You can see water molecules here that are hydrating those hydrophilic head groups. You can see there are very few water molecules that can make it into that interior. And you can see that it's not a rigid line. It's an approximate dimension. This is a non-covalent assemblage, and it's an average of a number of things. So it doesn't have the exact structure like a hemoglobin tetramer would. It has an average structure, and it has a dynamic structure. Fatty acids by themselves are not very common. We're going to look at them to start with so you can see what that component looks like, and then we're going to look at how we build them up into other things. Here are a couple of unsaturated fatty acids. This is oleate, and it gets its name from the fact that it comes from olive oil. It has a single double bond in it. This is a cis double bond where the other bond out of the carbon coming out of the double bond is coming out on the same side of that double bond. That makes it a cis. Cis always means this. Trans always means across. So this is a cis double bond. Here is linoleate, which comes from flaxseed. It has two of these cis double bonds. By themselves, those free fatty acids would not be very good for you. You're not going to find very many free fatty acids in your bloodstream. You're going to find them either bound to proteins, or you're going to find them as a covalent compound of various kinds. One kind would be taking a glycerol backbone like this and adding 3 chains to make a triacylglycerol. So glycerol is a good backbone for a lot of these things. It's a short 3-carbon piece, so it's not a lot, but it's large enough to pack things into something a little more complicated than fatty acids. And it has these 3 alcohol groups. And we're going to see that the fatty acids are going to be joined on through those groups. So instead of having 3 alcohols here, we have 3 esters. Here's one of the fatty acids that has made an ester here, and a second, a third, and so on. So this molecule now has no charged groups, and there is a bit of polarity here, sort of buried a little bit in, but not nearly as polar as a full fatty acid would be. We call this a tri-acyl glycerol because there's 3 acyl groups, ACYL, for the fact that you have a hydrocarbon side chain of some kind. Acyl is a very generic name. This is most commonly found in storage contexts, either in fat cells themselves or en route to them or from them. So we've talked about tri-acyl glycerols with the 3 fatty acid tails. A second major family of lipids would be glycerophospholipids. Now the glycero in there is telling you it's got a glycerol backbone, but these are now phospholipids. And the glycerophospholipids are going to be very important for membrane structure. They're not storage compounds the way the triacyl glycerides are. In the case of glycerophospholipids, you still have that 3-carbon backbone of glycerol, but you would only have 2 hydrophobic tails, which would be of some kind of fatty acid or other that's been combined in there covalently. And the third group is now going to be a polar head group. So, for example, phosphatidylcholine, here's the glycerol backbone. These 2 are the 2 of the fatty acyl tails. And here is a choline residue, and there's the phospho part that makes it phosphatidylcholine. Here is, again, the same glycerol backbone, 2 acyl chains. Here's a phospho group. In this case, it's ethanolamine. So there's an amine group there. You can see how that would have been ethanol if it had been an alcohol, but it's an amine, so it's ethanolamine. I'm not expecting you to memorize these structures, but walk through them with me so you can see how their names are telling you what they are. And they do have a general family resemblance through the glycerol backbones, the 2 fatty acyl tails, the phosphate group that's joining the glycerol backbone to whatever that polar group is. In this case, it's kind of like a twin situation. This is a glycerol as the head group, obviously polar there with a couple of alcohols left on it. And here is a serine, just exactly like an amino acid, except that the alcohol has been joined into the phosphate. Now, a very interesting effect of this is that while they're all amphipathic molecules, they all have a polar charged end or polar end with charges, yes or no, and very hydrophobic tails. They differ in the size and charge of their head groups. So here you can see there's 1 negative charge on the phosphate and a positive charge there. Overall, that is electrically neutral, but it does have charged groups. Here's ethanolamine. Same logic again, balance of charges, but actually charged groups. Here's phosphatidylglycerol. The negative charge on the phosphate, no charge over here, net negative charge on that molecule. Here's phosphatidylserine. And in this case, we have the carboxylic acid and amine group balancing each other out. So 1 net negative charge, but a lot more charge in general than some of the other ones. So these different groups can give a very different geometry and electrostatic cloud for membranes to work with. They're different in charge and in size. This is all important because the shape of lipids will determine how they pack. I mean, the shape of anything determines how they pack, but it's especially important in the case of lipids because that's what's going to determine the overall contours of the membranes. So if for instance, you have fatty acids, which have a carboxylic acid charged group at one end and then the acyl tail, the head is a bigger cloud than the tail is, so it doesn't really pack very well. It can slip onto the surface or into the surface of a protein, but it's not by itself going to pack very well. Here we have a glycerophospholipid as just kind of a schematic here. And now the two are in proportion, and so they can pack into a lipid bilayer like that. And then by contrast, if you look at a triacylglycerol, where you have a glycerol head group, which they haven't actually, I guess they're here, they're taking into account the other stuff besides the glycerol. And then you have your 3 acyl tails. The tails are going to be bigger in shape than the glycerol group, and now it can't make a proper bilayer, but it could make a droplet. It can make a fat droplet in a cell. So again, the structure will determine what higher order structures can form. Another big family of membrane lipids are the sphingolipids. There are a lot of these kind of odd Greek names in this group of things. I'm not quite sure why. Anyway, the sphingolipids have a sphingosine background. Now, here's the sphingosine molecule. Sphingosine has a what amounts to its own lipid built-in tail, but it has a double bond in there. This is, if you like, a 3-carbon backbone, sort of, with an amine hanging off of it and an alcohol there. This part in black actually came from a serine. This is the sphingosine backbone, and in a sphingolipid, this nitrogen will be occupied by another acyl chain. And so you can see over here, you now have a kind of a different looking thing than you would have had in a glycerophospholipid. And then it will have some kind of a head group, just as the glycerophospholipids did. This one has a choline group. So this is a phosphocholine up there, which we saw as a group in the glycerophospholipids. But this whole molecule with the phosphocholine there and this acyl group is sphingomyelin. And as you might guess from a name like sphingomyelin, it was originally isolated from nerve cells around the myelin sheath. And the sphingolipids play an especially important role in nerve cells. Any questions? Remember, if you have a question, there's probably someone else who does too. Okay? Let's more into the breach. Now, there's another molecule that has a very different kind of organization from the glycerophospholipids and the sphingolipids, and that is cholesterol. So cholesterol is also present in membranes. We're going to see it does a lot of things, not just membranes. But its overall architecture is radically different from the sphingolipids and the phospholipids. First of all, although we talked about the composition of the membrane as having amphipathic character with a polar part and a nonpolar part, this down here, whoops, sorry, I have to remember I can't click. This down here, that little alcohol, is the only polar part of cholesterol. Okay, that's its alcohol part, that little bit there. The rest of cholesterol is hydrophobic, but it's hydrophobic in a different way. Here is the ring structure of cholesterol. It's a fused ring structure, one double bond there, a couple of methyl groups hanging off. And then down at the other end, as far away from the alcohol as it can get, there's often a tail hanging off in the breeze. This part is obviously going to be somewhat more flexible than the flat ring part, but it's most of the molecule is this fused flat ring. There is another family of lipids that are not found in membranes. They're not structural lipids, but they do a lot of other things. And they're important because although they're not in all membranes, what they do is extremely useful in other ways. One of these families is called the isoprenoids. Here is the structure of isoprene with 2 double bonds there. It's a 5-carbon molecule. You may have heard of isoprene from its being used in making tires, I think. In any case, there it is. And if you string more than one of these isoprene units together, you can make polymers of isoprene that are then called isoprenoids. And they turn up in funny places. So here's ubiquinone. We're going to meet ubiquinone when we talk about electron transport. And it has a tail hanging off of it of 10 of these isoprene units. Here's phylloquinone. You can hear this quinone part, but that's referring to the ring structure. It's not referring to the isoprene tail. Phylloquinone has, again, this isoprene tail. It's vitamin K and is needed to do the modifications that allow for clotting. It's called vitamin K because it was discovered by Germans, and Germans spell clotting with a K. Here's retinol, which you may recognize as being an important pigment in the eyes. In this case, it's a multiply unsaturated isoprenoid where, because it's resonant like this, it has particularly good capacities for absorbing light. Another non-structural lipid is vitamin D. Vitamin D is not an isoprenoid. It's derived from cholesterol. You can sort of see here how that could have been a cholesterol molecule to begin with. And interestingly, it can be made from cholesterol by UV in the skin, that the energy of UV light is enough to lead to the photolysis of cholesterol and make vitamin D. There's a true and sad story about how in the Victorian period, women often developed broken bones towards middle age or a little later on because vitamin D plays an absolutely critical and irreplaceable role in calcium metabolism. And in the Victorian period, the women were wearing so much clothing that they had very little UV light on their skin, and they were having 6 and 8 and 10 and 12 kids. Where did the calcium for those bones and those kids come from? They came from their mom. And since they had very little vitamin D to get calcium to replace it with, every pregnancy ended up lowering the total calcium stores in the mother's body. So sunlight can be your friend, although nowadays we can get it as a supplement. Okay. So we've looked at these components as molecules, but they get a lot of their interest from the fact that they do come together to make lipid bilayers, and that these lipid bilayers have a dynamic structure. So here's a diagram that's a little more representative than the strict schematic that just has the wiggly lines. It's not quite as detailed as the ones that showed every atom. But it does convey to you the fact that the lengths of the tails and the shapes of the tails are going to affect how those lipid tails pack together inside the hydrophobic interior of a lipid bilayer. Now, this is a normal-looking fatty acid. This is a cis double bond here. Remember we talked about oleic acid as having a cis double bond. The normally occurring fatty acids in your body are all cis unsaturated double bonds in those fatty acids that are unsaturated. You can see over here in this diagram how a cis structure tends to make a kink, and that kink is going to contribute to the looser packing of that interior. In a trans fat, you have to imagine this being straight out, and now it's locked out straight out in that spot. So in effect, a trans fat behaves as a sort of a supersaturated fatty acid. It is less mobile than even a saturated fatty acid. In this schematic here, you can see the straight chains of the saturated fatty acids. They don't stay quite that rigid. They can wiggle around a bit. But the unsaturated fatty acids have this built-in permanent kink that makes them always be somewhat kinky, in addition to whatever additional flexibility they pick up from moving around just as thermal jiggling. These things come together in a number of dimensions. The longer a fatty acid is, the more rigid it's going to be, or the higher the melting point will be. The more energy it'll take to melt it. The more unsaturated a fatty acid is, the more fluid it'll be. It'll have a lower melting temperature. And interestingly, cholesterol has a behavior that's different from either length or saturation. So you can see in this particular diagram how there are some cholesterol molecules sort of intercalated here, sort of slipped in between the phospholipids. Presumably their little alcohol head group is just right out there peeking out. And cholesterol can act to buffer fluidity, if you like. Let me go back. By being a fluidity buffer, I mean that it will tend to, for instance, lower the melting point a bit compared to how a long fatty acid would be. It'll tend to raise the melting point a bit from an unsaturated fatty acid. So it'll tend to make the fluidity of the membrane vary less over a temperature range than that membrane would in the absence of the cholesterol. So what do we mean by saying that membranes are fluid, or fluidity in membranes? Well, the lipids, the fat part, are capable of considerable movement. If we look at lateral diffusion, diffusion from side to side in the plane of the lipid bilayer, in the plane of the membrane, we find that lateral diffusion is rapid. And they're trying to kind of capture that here with a little blue balloon that at this instant it's here and moments later it's over there. So side-to-side diffusion inside of one leaflet can happen quickly. However, if you think about transverse diffusion, moving from one leaflet across to the other leaflet, I think just a little bit of thinking will show you that that probably doesn't happen very often. Because for a phospholipid to diffuse by just random thermal motions from one leaflet to the other, that head group will have to go clear through that lipid interior, the hydrophobic interior. And that's not energetically favored. So transverse diffusion from one leaflet to the opposite leaflet is rare or slow. It's also called flip-flop because it's flipping from one leaflet down to the others. Transverse diffusion is accomplished through enzymes called translocases. So there are specific enzymes whose only job is to make sure that the lipid bilayer has the correct composition, and if necessary, it can actually flip phospholipids from one leaflet to the other. In addition to this extremely fluid part of the membrane, where there is really rapid lateral diffusion, we're discovering that there are also within that lipid bilayer, in living cells, not in synthetic lipid bilayers, patches of the bilayer that behave in a much more coherent way, things that are now called rafts. So these are a little patch of lipids that stay close to each other, that they stay together like a little iceberg floating in the sea. And their composition is different. This is just showing you a couple of the lipids that are typically found in these rafts, as well as some cholesterol. And so the composition of the rafts is different enough from the rest of the membrane, and the properties of those lipids are such that they don't separate from each other nearly as often as the rest of the lipids in the lipid bilayer. Okay. So let's take a moment. Here are three lipids. What would their relative melting points be? You know, again, melting point is like the TM for DNA. At a melting point, half of it is liquid, half of it is not. You can detect it through various parameters. So here we have three lipids. They're all C18. So what would we predict for these in terms of their relative melting points? Why don't you talk to each other to think a little bit about what you think? Hmm? Oh, okay. All right. Does anyone feel like you'd like to venture a suggestion as to which one of these has the highest melting point? Yeah? Good suggestion. You want to make a case for it? Why the transoleate would have a highest melting point? Okay, I'm going to repeat what you say because it can be hard to hear in this room. So the reasoning is like this, that a trans fatty acid bond behaves more like a saturated fatty acid or even a supersaturated. So the trans oleate will have a higher melting point than the cis. And the linoleate, coming from flaxseed, having 2 double bonds, will have a lower melting point still. And you can see this kind of amongst some everyday things in your kitchen. If you compare butter and olive oil and canola oil, for instance. The butter is going to be solid, more or less, at room temperature, depending on whether it's August or not. Olive oil is liquid at room temperature, but if you have fancy olive oil that you keep in the fridge, you know that in the fridge it solidifies. And canola oil, even if you put it in the fridge, will stay liquid because it has more polyunsaturated fats, multiple double bonds, than the oleate, the olive oil does. Any other questions about that? Okay. I guess I should have, I think this was before McDonald's claimed that they're going to take out their trans fatty acids. Some of you may know that I believe in New York City that they have made a ban on trans fats in their foods. It's pretty nasty stuff. Trans fats are formed from polyunsaturated fatty acids, that when you want to take something like canola oil and make something that's a little more solid out of it, like for margarine, one way of doing that is to hydrogenate it, bubble hydrogen gas through it with a catalyst, and some of the double bonds will turn into single bonds, but some of them will just be flipped. They won't be turned into single bonds. And so the trans fatty acids are only found in processed lipids. Now, we've been talking so far as if membranes are made out of lipids, and that's all you need to think about. But that would be a very misleading notion of membranes. Because membranes in cells, and actual membranes, not just lipid bilayers that have been made in the lab, membranes have proteins as well, and those proteins are very important. A typical membrane will have about half of the mass be traceable to proteins. So the barrier function of being able to exclude stuff is due to the lipid bilayer. The self-healing character is due to the lipid bilayer. The spontaneous assembly is due to the lipid bilayer. But the rest of the stuff that membranes do can only happen if there is some protein in it that can confer those functions on it. And we talk about proteins that are associated with membranes as falling into several classes. There are some membranes that completely span across the membrane, so they have some exposed on the far side and on the near side. These are considered integral membrane proteins. They're integral to the membrane. They are completely a part of it. There are peripheral membrane proteins, which are frequently found on those membranes, but that are only associated at the surface. Now, functionally speaking, as opposed to other kinds of structural information, an integral membrane protein will only come out of the membrane with fairly strong detergents. As you can imagine, you have to actually foam up those lipids and get them off. By contrast, a peripheral membrane protein can often be taken off by using high salt or alkali that you can compete for or interrupt or disrupt the electrostatic interactions at that interface and remove the peripheral membrane proteins without detergents. In fairly recent years, we've discovered a third group of membrane proteins that gave rise to some confusing diagrams in older textbooks. And these are membrane proteins in which all the protein part is outside the membrane, but it has a covalently attached lipid tail, and that lipid tail can then penetrate into the lipid bilayer as an anchor. Now, you can imagine that this by itself, salt won't get it out, but a gentler detergent often will. So this is a lipid-linked protein. In terms of the exposure of its protein parts, it would behave like a peripheral membrane protein, but its lipid tail affects how easily or difficultly you can get it off the membrane. Now, to make a membrane protein, an integral membrane protein that goes clear through the membrane, there are a couple of ways of doing that. One way of doing it is by using multiple alpha helices. And so here's a diagram. This is showing you a skeleton diagram of the alpha helix. Here's a ribbon diagram. And you can get a sense, I think, here for the way in which you can have several of these alpha helices sort of forming a bundle, if you like. Little bits of the protein may be exposed outside the lipid bilayer, in some cases quite a bit more than this little bit. This particular membrane is one that's at the cell membranes. They're calling that the extracellular space. But you can imagine that one of the important things in this case is that the side chains in this part of the lipid bilayer are themselves fairly hydrophobic. And you can see in this particular case, you've got some phenylalanines, you've got what looks like a leucine here, it looks like a tryptophan. You've got a variety of hydrophobic amino acids that are sticking out. They're sticking out of off the surface, if you like, of the alpha helix and making contact because they're hydrophobic. Given the dimensions of an alpha helix and the dimensions of lipid membranes, it takes about 20 residues to go from one side to the other. So about 20 amino acid residues in a row have to participate in that alpha helix for it to be long enough. And as a consequence, we tend to see things that are kind of characteristic of these membrane-spanning things. They're not absolutely characteristic, but there certainly is a strong tendency. If you look at this particular alpha helix that is a membrane-spanning alpha helix, what you'll see in here is that once you get past this glutamate, you've got tryptophan, isoleucine, tryptophan, leucine, alanine, leucine, glycine, threonine. Not a very hydrophobic thing, just that one little alcohol. Alanine, leucine, methionine, glycine, threonine, another little alcohol there. But this is a fairly, then at the end here, you've got a lysine. So you can see at this end and at that end, you've got some charged things. In between, you have things that are only hydrophobic or only weakly polar, like a leucine, like a threonine. And that's a typical kind of transmembrane sequence. Another strategy for spanning the membrane in an integral membrane protein is to use beta barrels. And this is a configuration that I don't think we've seen yet, in which you have beta-pleated sheets that are lined up side by side to each other, but with a little bit of a twist, so that each beta sheet is just a little bit around the other way. And when you do that, you can pack an entire cylinder of beta-pleated sheet. And these are often called beta barrels, because if you've ever seen a barrel, it has that general shape to it. To go clear around, you need at least 8, or you're not going to get far enough around. But they don't have to be just that. You can have a looser packing up to 20 strands. And in the case of a beta barrel, that the transmembrane portion is not as distinctive as it would be with an alpha helix. For one thing, beta sheet is more extended, so the number of residues that are in it is not as long. But secondly, it's only the residues that are facing out into the lipid bilayer that need to be hydrophobic. There may well be residues pointing towards the interior that are polar or even charged, depending on what the function of this membrane, this protein is. There are often aromatic residues at the edges of this. You can see here this kind of frill or collar of aromatic side chains there, and another one down here. And you can imagine, the bulk and size of aromatic side chains. Those are very hydrophobic groups that they act as a way to keep that thing in the lipid bilayer. I've already answered my own question. I'm sorry. So there we go. In terms of the lipid-linked proteins, that third class I mentioned in there, there are several kinds of lipids that can be added, and there are several ways of adding them. So, for instance, you can have a myristyl group, a palmitoyl group, a prenyl group. Here you can see, if you remember back to when we were talking about the isoprenoids, this looks like a cousin to the isoprenoids. This would be a prenyl group here. I'm just guessing here. I would guess that this is probably myristil and this is probably palmitate, but don't worry about it. Myristil is shorter than palmitate. There are some that are linked through a structure that's actually has sugar in there as well as the fatty acid tail itself. You can see here that this particular side chain has been added onto a sulfur that's at the end of a protein that was also methylated. So this particular one has a prenyl side chain and a methyl group to cap that oxygen so it doesn't have a charge there. Here's one that's been attached to a nitrogen that's internal to the chain. Here's one where it's been attached to a sulfur but internal to the polypeptide. So a number of different strategies for adding these tails to things. So overall, what's the relationship between the lipid part of a lipid membrane, of a lipid bilayer, of a membrane, and the protein part of a membrane? You've heard the phrase probably before of the fluid mosaic model. And I'm going to corrupt your minds with the notion of a solid mosaic model for at least part of it. Now, the fluid mosaic model is one that proposed that although there's a sheet of stuff there, that the things in it can move side to side like a lipid sea. And it is true that lipids, for the most part, diffuse freely in the plane of the membrane. Remember, we talked about lateral diffusion being fairly rapid. They don't diffuse across, but they diffuse in the plane of the membrane pretty well. This doesn't work nearly as well for proteins. The very earliest studies of membrane structure were done in red blood cells, and the very earliest probes were, in fact, lipid probes, so no surprise there. And even when they did start using protein probes in red blood cells, the way they treated the red blood cells at that point, they weren't nearly as tough as they had been, and red blood cells aren't as complicated as real cells anyhow. I hope there are no red blood cell lovers in the audience that feel offended. So what do we see if we look more specifically at individual proteins or individual classes of proteins in actual cells that are alive? There are some proteins that do not detectably move. That if you can label by one means or another, that they're here when you start the experiment, they're here in 10 minutes, they're here in 2 hours. So there's some membrane proteins that are simply not going to go anywhere. There are other membrane proteins that move around, but in an area like this. And for both of those, we have information that suggests that those behaviors are due to the fact that these are proteins that can interact with the cytoskeleton one way or another. This is just a diagram that's trying to give you a sense of that membrane structure. Here's your lipid bilayer part, and you can see why there would be free movement. This is showing you the membrane proteins, but this particular picture is not showing you anything of the other stuff inside, which I'll have pictures for in a second. One of the things that this picture is starting to highlight is the yellow stuff on this is all sugars. It's carbohydrates of one kind or another, and we'll talk about that more later too. There are, in some cells, some proteins that can move. They don't move at the speed of diffusion, but they can move, and they may well move in a fairly broad distribution across the cell. These are pictures that are not found in your textbook, so if you're watching this on the video, please pay attention. And they really highlight the fact that there is an element of the cytoskeleton that's in intimate contact with membranes in almost every cell examined. Remember we talked about the cytoskeleton back when we were talking about proteins. This is a place where the two are actually kind of interacting with each other. This particular picture is showing you some of the kinds of contacts that are common in cytoskeleton membrane contacts. It's drawn the way it is and can show you, for instance, This is an ion exchanger. This is a transport molecule that's anchored to the membrane cytoskeleton. These are other molecules that are anchored to the cytoskeleton. What it does not capture is the density. That the density of proteins in the membrane and the density of cytoskeleton underneath it are anywhere from 20 to 100 times greater. If you think about protein being half the mass of the membrane, when you look down on top of it, it should be almost like a checkerboard that the white squares are lipid and the black squares are protein. It's that dense. So our normal pictures of it in our mind as being mostly lipid can be misleading. This is a clever method where they were able to tag membrane proteins with, I think, a gold particle in this case, and could track individual molecules over time. And you can see how there's this motion within an area, but not just sort of going anywhere at all. This is a diagram that captures, again, the sugar part. Here you can see the extracellular sugar, carbohydrates here. This is one of the few I've seen that incorporates cholesterol molecules to try to give you a sense of how that ring structure is actually longer than one leaflet of a bilayer. And it's reminding you of the fact that there are receptors anchored to the cytoskeleton as well as transporters. There are a lot of places where it's hard to capture the real density of things. I just mentioned now the density of proteins. The density of sugars on cells is, if anything, even harder to grasp. The sugars in cells in general are going to be the sugars on proteins or on lipids will be on the outside of the cell. So with glycolipids, glycolipids are lipids that have, in addition to the acyl side chains, they have sugar groups, sometimes quite complicated ones on top. Those sugar groups will face outside. Glycoproteins have, again, derivatives on them. Those are, again, facing outside. And this picture captures for you just how thick that is. So this is a picture of a cell. This would be the membrane right here. It's a very thin thing compared to the rest of this. And it's been stained with something that highlights the carbohydrate. And I don't think most of us walk around with the notion of the exterior of cells that looks like this, that the carbohydrates on the outside of cells are almost like those thermofoam mattresses, that there's almost a buffer around every cell of carbohydrate. Some of the places where that can be important are on the blood cells. And this is the carbohydrate is behind the whole thing of type A and type B blood. So if you want to symbolize this somewhat schematically, you have either lipids or proteins. In a person who's type A, both lipids and proteins will have some of these derivatives. In this case, they've marked the characteristic sugars by color. So type A people derivatize their proteins and put the blue one on. Type B put the purple thing on. Since type B people have the purple thing, they can raise antibodies against type A. And since the type A people have the blue thing, they can raise antibodies against type B. But people who have blood type O don't have either one of those. So someone who's type A or type B can receive type O blood because there's no nasty sugars on it. So type O's are universal donors. You can imagine that someone who's heterozygous, has both type A and type B, now can't take blood from anybody except from a type O or a type AB. Rather, they can take it from everybody. They're universal recipients. They can't give it to people. Okay, well, it is time. I'll see you then on Wednesday. And try to be safe out there. The weather isn't looking as bad as it did a day ago, but it still doesn't look nice.